You had questions, and I have some answers. First, the number one most asked question that I get: where are all the other people in my lab? I swear that I do have coworkers and lab mates, but I also want to respect the fact that this is the place where we work. So I try and film on nights, on weekends, when there aren't other people working. Now, that's not to say that people don't work nights and weekends. Both of those things happen in the lab, but I try and find times where there aren't people here because I do want to respect that this is a workspace, and me sitting over here talking to my camera would probably be very distracting from someone actually trying to do work. I swear I don't work in a ghost lab. Arch of Winter asked about how my colleagues and professors feel about me making these YouTube videos. Do they know? Is there any pressure by them to change how I'm doing them? And if anyone has asked to make a cameo. So my lab is thankfully very supportive of me making these videos. I know that some of my lab mates watch them sometimes. I know that my PI watches them sometimes. And so, you know, they're aware and they're very supportive. I'm sure there are probably a couple people in my department who are rolling their eyes at me, but that's okay. So far, everyone I have encountered has been very supportive. There's no pressure for me to change how I'm doing them. Everyone seems okay with it. And I have reached out to some people to maybe start asking for some cameos because I'd love to show you some other people in my school and department and lab, but not everyone is super comfortable being on camera and nobody has asked for a cameo yet, but they should. Naughty Ninja Bears asks, what is your favorite thing about working in a lab and your least favorite thing? Starting with my least favorite thing, there are definitely some things in lab that are just super tedious that you have to do over and over and over and over again. Whenever I do a bunch of RNA or DNA extractions from a bunch of samples, I have to go over to the nanodrop, a little machine that helps me calculate all of the concentrations of the DNA or RNA in those samples. And I just can't stand nano dropping recently. You just stand there, you take a sample, put it on the nano drop, hit the button, take a sample, put it on the nano drop, hit the button, take a sample, put it on the nano drop, and hit the button. Oh my gosh, my brain just doesn't want to do that. So there are a lot of those little tedious tasks in lab that I feel like I go through cycles of which one is my least favorite, and currently nano dropping is my least favorite thing. It's so boring. But, you know, you gotta do it. On the other hand, I think my favorite thing about working in a lab is that it's pretty active work. I'm not sitting at a desk all day. So while there is a lot of reading and writing component to doing science, because I'm doing wet lab work, there's also a lot of standing up and walking around and doing stuff at my bench and then doing stuff in the tissue culture hood and then going over to this place and getting a sample from that place. And so it's sort of a nice active thing where I'm not stuck at a desk for 8 hours a day, and I super appreciate that. I really like the activity of doing lab work. Valentine Molchanov, I'm so sorry if I mispronounced that, asked, why did you choose specifically genetics and not something like physics? That answer is quite long, but the condensed version of it is that I love biology, and I feel like you can use genetics to interrogate most parts of biology, and to me, I know I'm a geneticist and I'm biased, but most cool biology boils down to some cool genetic piece. And so genetics to me was the key to open up all of biology. And I think one of the reasons why I really wanted to study it in my PhD was because I was so fascinated by the fact that we now have tools to control our DNA and our RNA and really control our genetics. And when I applied to grad school, this was pre-CRISPR times, right? So I was really super excited about RNAi and different methods that we could use to turn off genes. But now, like, the field has exploded and we have all these different ways to edit genomes and to turn genes on and off at all of these different levels, and I just find that to be amazing and so cool, and I'm excited that I'm able to sort of play around in this field at this time when all of those tools are exploding. Rudius Greyrat asked a bunch of questions, but the one I'm gonna pick is, what is your manifesto for this channel? It's a good question. I've certainly never... No fridge! Oh! Can that be my new form of like swearing in these videos? Because I don't swear in these videos, but if I wanted to, instead of saying another F word, could I just be like, fridge? 'Cause fridge. I've certainly never sat down and created a manifesto for this channel, but my hope, my goal is to humanize science. And so I actually stuck a little note up above my computer at home recently that says, tell science through human stories. And I don't know that that's the manifesto for this channel so much as I want that to be the manifesto for my career, but I'm hoping that this is certainly the first steps of that career. So maybe I need to be better at also here telling science through human stories. On Twitter, More Robots asked, do you see science robots, automated testing and procedure, expanding your field or denying undergraduates critical experience? No. You see, undergraduates are so much cheaper than automated robots. We will always use the undergraduates. Seriously though, I do imagine that some amount of automation will help the field, but often that automation happens in industry and not in academia. So in industry, they can buy a robot to change media in all their cells every day. In academia, a graduate student is much cheaper. James Lloyd asked, what is a nice thing your lab does that not all labs might do that helps you, like providing racked sterile tips or pot washing? So our lab does both. You probably want something more than that we buy sterile racked tips and that somebody else washes our dishes, but people do both of those things and it's super useful. I have in the past had to rack my own tips and then autoclave them, and yeah, you can do it and you save money doing it, but oh my gosh, it's so much easier and nicer to just buy the ones that are pre-done. Carly Ann asked if I'll be doing a video about the genetics of tea. Oh my gosh, I hadn't thought of it, but I totally should. Dan asked, where did I go to school for undergrad? I went to Brandeis University outside of Boston, and it was pretty awesome. Claire from Brilliant Botany asked, what is your favorite part of being a sci-commer? And it's absolutely just getting to talk to people about science. I mean, I love talking about science, and doing that in a science communication standpoint reminds me why I love science. And so I think that's important sometimes when you're in the lab and stuff isn't working and you're like, why am I here? This is bad. It's nice to have an outlet where I can remember like, oh, because it's cool, like science is great. And so getting to do science communication helps me remember the cool stuff about science. Finally, James Gurney asked me, 100 duck-sized horses or one horse-sized duck? And I know that this is supposed to be like they're fighting each other and who's gonna win, or like I'm fighting them and like which one would I rather fight, but hold up, 100 duck-sized horses? Oh my god, I wanna play in a field full of 100 duck-sized horses. There is no fighting here. There is just 100 tiny horses running around in a field. That's a great way to end a science video, right? I did not get through everybody's questions, but I will probably make another one of these in the future, so if I didn't get to your question, you will have an opportunity to ask it again. Go forth and do science. What is this thing? It's heavy is what it is. It's used for mixing things.